Yeah, the way we think about the biological research workflow, in recent years there's been a lot of interest in the automation. In fact, I was actually one of the early members of another MIT company named Ginkgo Bioworks that has built much of the early tools for automation in molecular biology. And I built a lot of the automation systems then. So I built a lot of knowledge understanding what automation was good for and what it was bad for. And I think that, that there's a lot of interest in automation, and there should be. However, it's not the whole picture. One trend that we've seen with the automation is that it's mostly driven by computer scientists who have the tools, but who don't necessarily know the intimate problems of being in the lab, for long hours at a time. They're not biologists. And so Biobright kind of inverted that. We are biologists who've learned computer science and electronic engineering, so we could solve those problems with very intimate knowledge of the workflow. And that's why I talk about the necessity for human augmentation, putting the human at the center of the loop, not taking them out of the loop like automation does. But those two things go hand in hand. Automation is excellent at doing workflows and protocols 1,000 times over, but it is very brittle. It has no ability to adapt on the fly. It has no resilience to changes. Research really, for the most part, is an exploration, which means you are changing parameters on the fly. You are adapting. You are deciding to ignore particular changes. And research often is at the beginning, before development, before scale up, even in pharmaceutical environments. Research always happens first. And we found that most of the time and most of the reproducibility problems actually come from translating that workflow from one person to another, from one group to another, from even a geographical location to another. And there are very few handles on that. Automation will not help with that. Human augmentation, on the other hand, allows you to provide almost superpowers to the scientist, being able to, at a glance or with a voice command, say, Darwin, show me the average temperature that I've done this step at for the last 3 months. Or show me how Mike did it last week, and pop a video. So the way the Biobright platform works is that we centralize the information onto our systems, and then you can authorize specific users either in your lab or across your organization to highlight and review the information that you've done. So if I were to do a protocol, say here in Cambridge, Massachusetts, and it's the dead of winter, it may be very different from doing the experiment across our organization, say in San Diego, California, at the same time of year. It may be very important, it is very important for the scientist at the other end who is going to take that experiment to be able to see that information. That happens through our platform, and we are exploring ways of how to make that as easy as possible, potentially even with just an attachment to an email, etc. The access to the platform allows you to see all of that with the permissions, obviously, that you require. But there are other ways that we are looking into to augment the relationship so that people don't have to change their workflow. This is really key. We don't want people to change their workflow in the way they share their experiments.